Good morning, students. Myself, Babida Matthew. Today I am going to discuss about immunology in obstetrics. What is immune system? What is immune system works in our body? So when an antigen comes to our body, it acts as our antibody in our body. It acts against the antigen and keep our body immune and prevent the external stimuli or the microorganism. Okay, that is the immune system work in our body. So we can see the basic immunology of pregnancy. One tissue that is repeatedly grafted and repeatedly tolerated is the fetus. The mysteries of mechanism of the immune system that prevent rejection of the fetus that remain unknown to the immunobiologists. We can see the immune system. We can see it is classified into two arms. First is innate immunity and second is adaptive immunity. Each one we can see in detail. The innate immune system. It is the first line of host defense against infection and it works fast once it recognizes the pathogens. But it cannot identify the self but non-self. The immune cells involved are phagocytic and natural killer cells. So which is phagocytes? Phagocytic are neutrophils, monocytes and macrophages. Natural killer cells are eosinophils and basophils. The immune response are by the release of cytokines and chemokines. So when the antigen comes to our body, it responds by the release of cytokines and chemokines. Phagocytic cells ingest and digest microorganisms with the lysosomal enzyme. The NK cells or natural killer cells can recognize and distinguish between normal cells and cells infected with virus or tumor through the expression of MHC class I antigens. The NK cells destroy cells that are deficient in human leukocyte antigen class I molecules. Adaptive immunity is the second line of immune defense. It prevents reinfection through the immunological memory. Next we can see what is acquired immune defense. It is contributed mainly by two types of cells, that is lymphocytes, that is B and T lymphocyte, and antigen presenting cells, that is APCs, antigen presenting cells. The T lymphocytes are classified again into T helper and T cytotoxic cells. The cells have two subtypes, T helper 1 and T helper 2. T helper 1 cell activate macrophages and are involved in cell-mediated immunity. But T2 cells activate B cells differentiation and are involved in the humoral immunity. When pathogens or virus replicate inside the cells and are inaccessible to antibodies, are destroyed by the T cell. This is the acquired immune defense. Next we can see the major histocompatibility complex, that is MHC, major histocompatibility complex. It is a complex of genes with multiple loci. It is located on the chromosome X, and MHC or the major histocompatibility complex encodes two types of membrane protein. Those are known as human leukocyte antigen or HLA class I and HLA class II. The process of graft rejection generally involves recognition of foreign MHC molecules by host T1 and lymphocytes. So, if any graft in our body that recognizes MHC molecule by the host T1 and T1 lymphocyte. Next, we can see the immunology in pregnancy. Pregnancy is not an immunodeficient state. Women are able to respond to both humoral and cell-mediated immunity against the paternal antigen. Paternal means a father antigen. A specific type of NK cells, natural killer cells, that is large granular lymphocytes, are present in the decidia. So when the fetus is formed, or after the fertilization, the fertilized ovum is implanted in the decidia. So when the decidia formed for the implantation, the natural killer cells are present in the decidia, mainly at the site of implantation. The NK cells are different from the blood NK cells. So uterine NK cells are different from the blood NK cells. These uterine NK cells control the trophoblast invasion when they interact with the trophoblast HLA class 1 molecule. The uterine NK cells depends on progesterone for its survival. So, when uterine NK cells contribute to maternal tolerance of the fetus and maintenance of pregnancy. So, that's why it is not rejected. Then NK cells share many antigenic and functional similarities to T cells. There is high, or that is 30% proportion of NK cells in the fetal circulation up to 13 weeks. Due to their high number, early presence and the ability to kill cells, it is likely that these NK cells are very important in fetal innate immune system. The uterine macrophages are the major source of nitric oxide and prostaglandin. Next, trophoblast cells are derived from the fetal tissue and invade the decidia when it is implanted. This trophoblast is developed as the placenta. These trophoblast cells forms the interface between the fetus and the mother. Thus, the placenta forms an efficient barrier against the transmission of immunocompetent cells between the fetus and the mother. So that the trophoblast cells help them to prevent the transmission from the maternal to the fetus. The trophoblast covering the chorionic villi comes in contact with maternal blood in the intervillous space and interact with the maternal systemic immune response. It does not express HLA class 1 and class 2 molecules. The trophoblast in contact with the decidia. So when after the fertilization, it is implanted in the decidia. The trophoblast, it is contacted in the decidia. So it expresses HLA class 1 molecules but not HLA class 2 molecules. This interacts with the maternal local uterine immune response. Placenta presents no immunocompetent cells due to the lack of HLA antigen, and placenta act as an efficient immunological barrier. During the pregnancy, maternal immune response is shifted from T helper 1 to T helper 2 type, and Th2 type response is beneficial due to the production of cytokines. Deviation results in improvement of woman with rheumatoid arthritis in the pregnancy. So during pregnancy, the placenta helps as a barrier between the mother and the fetus. During pregnancy, there is a production of antibodies of the paternal antigen. These are anti-HLA antibodies and antibodies against sensitized T cells. These antibodies have no major effect on the pregnancy outcome. Immunological mechanisms involved in the pregnancy are not the same as that of organ transfer. So, when an organ is transplanted into the body, that is also external or a external thing. But it is not. So, if the organ transplanted, there is a chance of rejection. But the same mechanism is not involved in the pregnancy. Pregnancy also is a foreign substance. It is implanted into the uterus, but it is not rejected. So immunological tolerance through complement and cytokines regulation is protective for the pregnancy. That's why the pregnancy is not rejected. The maternal fetal HLA incompatibility is found beneficial in human pregnancy, whereas HLA compatibility often results in miscarriage. If the HLA incompatibility occur in the human pregnancy, there is a chance of miscarriage. And endometrial T cells have immunosuppressive property. Next is the ABO, hemolytic disease of the newborn. This is also immunological problem. So, in this, jaundice in newborn infants within 24 hours of pregnancy, it is due to the ABO immunization of the mother. The incidence is higher in group O mothers carrying the group A1 fetuses. Except for the fact that A1 antigen is strong, it has not been clearly known as to why the A1 fetuses mostly take the blood. It is postulated that IgG anti-A, anti-B are formed more commonly in group O mothers. So, IgM anti-A, anti-B maternal antibodies are also known to play some role in bringing about ABO, hemolytic disease of the newborn. Next is RH isoimmunization. Though entry of the fetal blood in maternal circulation can take place at any time during the pregnancy, because if there is any pre-core injury into the placenta, and after the separation of the placenta. So, this time the fetal blood may enter to the maternal circulation. So, fetal maternal bleed is common in the third trimester, particularly during the separation of placenta, or I already explained due to some invasive procedure. So, 0.1 ml of RH positive fetal blood is sufficient to bring about immunization in RH negative mother. So, immunization against RHD antigen is most frequently as D is more potentially antigenic than the other antigens like C, E, etc. So, fetal RHD positive red cells gaining entry into the circulation of a RH negative mother takes several weeks to immunize her. So, under these circumstances, the mother will form anti-RHD agglutinin which will pass again through the placental barrier into the fetus, giving rise to the agglutination or hemolysis of fetal erythrocytes, which ultimately may lead to dangerous situations like hydros fetalis, icterus gravis neonatum, or kernicterus. That is the RH isoimmunization. Then pre-eclampsia and eclampsia. In this condition, anti-trophoblast antibody are found in the maternal serum. It is known that placental and renal antigen cross react. So, when the placental antigen and the renal antigen cross react, that's why the pre-eclampsia is occurring. Renal lesions are common in the gestosis. There are subendothelial deposits of immunoglobulin, particularly IgG and IgM, fibrin and complement, suggesting that this is an immune complex disorder. Further observation are exposure to sperm from the reproductive partner or exposure to placental or fetal cells is predictive. Then preeclampsia is high in the pregnancies with sperm donation. So donation or oocyte donation, as there is no self-antigen sharing. Then interaction of HLA class 1 molecule from the extracellular trophoblast with the decidual NK cells regulate the trophoblast invasion. So because of these reasons, there is a chance of preeclampsia or the eclampsia because it is an antigen. Next, spontaneous or recurrent abortion. Why the spontaneous or recurrent abortion occur? This is also due to the immune response. There are some observations suggesting an immunological interaction in cases of spontaneous and recurrent miscarriages. Generally, The ABO group of mother is different from that of the fetus, and incidence of abortion is higher than when fetus is of group A, then mother group O, and the father is group A. And recurrent aborters reject skin graft from their husbands earlier than from the unrelated males. Following a change of husband, recurrent aborters are known to have successful pregnancies. Successful pregnancies generally dissimilar concepts and adequate T2 maternal immune responses. Fetal HLA phenotype must be different from the mother to generate the T2 immune response. Woman with recurrent miscarriage produces low level of T helper 2 cytokines. So, the woman with the low level of T2 cytokines have the more chance of recurrent miscarriage. And women with recurrent miscarriage share more HLA antigen with the husband than the couples with a successful pregnancy. So, more antigen is sharing to the woman. Those who have recurrent miscarriage, usually in the pregnancy, from the paternal to the maternal, antigen is sharing, but it will be more when the miscarriage occurs. Women with recurrent miscarriage have the decreased population of NK natural killing cells. When the woman is have a decrease in the decidua, there is a chance of recurrent miscarriage recently occur. This indicates an altered immune environment within the decidua. The regimen of immunotherapy to women with recurrent miscarriage is based with the idea to stimulate the immune response against her conceptors, and injection with the husband's lymphocytes have been tried, but there is no conclusive evidence of its benefit. So, because of this, husband's antigen is it is not reacting properly by the decidua, this miscarriages occur. So, the studies shows that injection of husband's lymphocyte to prevent the miscarriage, but not much effective is seen in this method. Then antiphospholipid syndrome. In SLE, two antiphospholipid antibodies, that is lupus anticoagulant and anticardiolipin, are important. These antibodies act by the dysregulation of the coagulation pathway. This causes thrombosis of the uteroplacental vessels. So when the dysregulation of the coagulation pathway affected by these antibodies, that causes thrombus formation in the uteroplacental vessels, and so that causes the poor placental circulation, and that also furtherly lead to the obstetrical complication to this pathology. Next, maternal autoimmune disease and the fetuses. The incidence of neonatal thyrotoxicosis is higher in babies born of thyrotoxic mothers, and a baby to a mother with the ITP will in all probability suffer from the same disease with the transplacental transfer to the antiplatelet antibody. So, the same mother having, if there is a mother is having thyrotoxic problem, it can transfer through the placental barrier to the mother and to the fetus, and baby also suffer from the neonatal thyrotoxicosis. Next, Myasthenia gravis also have some such relationship due to the transplant. In Myasthenia gravis also, if the mother is having that, it can also be transferred. Transplants then will transfer to the fetus or transfer to the transfer of acetylcholine blocking factor, and babies born of mother suffering from the systemic lupus erythematosus often develop congenital heart block due to the transmission of anti-RO and anti-LA antibodies. And SLE patients very often have exacerbation of disease activity during the pregnancy or in the early postpartum period. All the diseases listed in this group manifest transitionally to the newborn. So till now what we told thyrotoxicosis, it can be transmitted, and myasthenia gravis. So these diseases can be transmitted to the baby by the transplacental transfer. Next is immunological control of fertility. First is the A is the placental hormones. Human chorionic gonadotrophy that cross reacts with the luteinizing hormone and thyroid stimulating hormone and follicular stimulating hormone has the beta subunit, that is non-immunology, so as to be coupled with the HAP10-4 antibody production. So, HCG can be detected on the trophoectoderm as early as the blastocyst stage prior to the implantation. Thus, the anti-placental antigen has been focused on the anti-HCG vaccine. Of the many anti-HCG vaccine, most effective being the one that is directed against the C-terminal peptide on the beta subunit of HCG. This antibody does not cross-react with the LH and thought to be effective up to 12 months. That is a placental hormone. Next is a non-hormonal trophoblastic antigen. Anti-sera against two trophoblastic proteins, that is SP1 and PP5, are being prepared in the experimental animals. So non-hormonal trophoblastic antigens are anti-sera. Anti-sera means that is antibodies against the specific antigens. Okay, so that produces two trophoblastic proteins. That is, one is SP1 means C1 George Church protein 1 and placental protein 5. That is a 2 are being prepared in. It is tried in the animal experiments. Third one is human sperm antigen. This is all causes. Now we are telling that these causes are these reasons are causes the fertility. Infertility. That is a control of fertility, because of this immunological control of fertility means fertility is not occurring because of these immunological factors. So that's the first when we told placental hormone. Second is non-hormonal trophoblastic antigens that is present in the body, and third is human sperm antigen. So these of the numerous sperm antigens usually enter to the body when the sperm is entering to the woman's body. So antibodies against the LDHX is being currently evaluated, and there is a significant reduction of viable sperm as well as improper implantation of the ovum is experimental animals done. So when anti-LDHX antibody is injected into them, there is some result, that is good result has been shown. So because of anti-sperm antibody, there is a chance of infertility or control of fertility. Next is zona pellucida. Zona pellucida, it is a covering. Okay, so zona pellucida, so covering is zona pellucida. So zona pellucida is the most extensively studied potentially oocyte target antigen. So anti-sera2 ZP blocks sperm penetration as it coats the zona surface. So sera from infertile women with anti-SP antibodies can block in vitro fertilization, and efforts have been focused on developing a zona pellucida immunogen with only B cells episode. So if there is only B cells, so there can be no rejection. This approach has been successful in inducting long-term but reversible anti-fertility effects is experimental, only done in the experimental animal. Then next is local immune response of the genital tract also causes the infertility. The endometrium and vaginal mucosa does not secrete IgA. So the atoms are being does not secrete the IgA. So that also causes the infertility. So atoms are being made to produce local immunity. So if there is local immunity in the is produced in local immunity in the cervix against sperm antigen by stimulating the production of secretory IgA. So if it produces the local, it is a sperm is an antigen. So when there is a local immunity produces, that is if it does not allow the to enter into the uterus. Okay, so that is the local immune response of the due to the local immune response, that is in the vaginal mucosa and endometrium, there is a chance of the infertility. Then anti-sperm antibodies, that is the IgG, IgM and Ig are the antibodies. Okay, that is anti-sperm antibodies are present either in the serum or the reproductive tract. So if the woman's reproductive tract, these anti-sperm antibodies are present. These antibodies, antibodies present in the reproductive tract, the antibodies present in the female reproductive tract that bind the sperm surface antigen. So in the sperm there is an antigen. So it binds the antibodies in the woman reproductive tract that bind the surface antigen of the sperm and affect the motility of the sperm. So it cannot properly move. The sperm motility is affected and the sperm cannot move properly to the uterus. So that's why it causes infertility. That is the anti-sperm antibody. So that's all about the immunology in obstetrics. So till now we discussed about the immunology in obstetrics, what is the basic immunology of pregnancy, and it is two types of immunology are the, that is the innate immune defense and acquired immune defense. And we discussed about the immunology in pregnancy and ABO, hemolytic disease of newborn, RH, isoimmunization, preeclampsia and eclampsia, how it occurs due to immunological problem, and spontaneous or recurrent abortion due to this immunological response, and antiphospholipid syndrome, maternal autoimmune disease of the fetus, and finally we explain about the immunology control of fertility, that is what are the factors what causes the infertility due to immunological problem. That's a plasmal hormones, non-hormonal trophoblastic antigen, human sperm antigen, zona pellucida, local immune response of the urinary tract, and anti-sperm antibodies. That's all about today's class. I think all of you are interested.